I'm Ria, and before we go into how firefighters, potions, and English essays relate to gene editing and treatment of hemophilia, let's talk about how you're doing, and more specifically, how you started your day. Maybe you walked downstairs and made a cup of coffee and some breakfast. Or maybe you typed up a to-do list for the day and sent out some emails. Or maybe even went on a walk or run. And all of these activities seem normal, but everyone with hemophilia can't do these things every day. Hemophilia is a single gene disorder, meaning that all of the complications that arise with this disease are caused by a single mutation. There are thousands of possible mutations, but in order to try and help the most people and the abundance of research already conducted, I chose to focus on the most common mutation of hemophilia A, the inversion 22 mutation on the F8 gene, which accounts for almost half of all hemophilia A cases. As some background, hemophilia is a recessive X-linked disorder. And you may have learned a little bit about genetics in a high school biology course. Sorry for giving flashbacks to drawing numerous Punnett squares on tests and decoding passages about Mendelian pea plants, but these ideas about inheritance are actually super important in this case. As you can see on this diagram, men are more affected than females for hemophilia because of the inheritance pattern. Hemophilia is a recessive X-linked disorder, and since females have 2 X chromosomes and men have 1, if men have the gene, it is expressed and they have hemophilia, but females are biologically lucky here, as if they have 1 gene for hemophilia, they can still have a chance for the gene to be overshadowed by a dominant normal gene. And if they only have 1 hemophilia gene and 1 normal gene, they're carriers, meaning that they don't have the symptoms, but they can pass the gene on to their children. In hemophilia A patients with the inversion 22 mutation, exons 1 to 22 out of the total 26 are reversed, which alters the sequence of base pairs and therefore the encoded protein. When this occurs, a protein called coagulation factor 8, which is the main protein in the process of blood clotting, is not produced in sufficient amounts. People with hemophilia often have a lot of internal bleeding and swelling as their blood doesn't clot as easily, which can cause a lot of symptoms that are painful and life-threatening as the hemophilia becomes more and more severe. The factor 8 protein essentially works in your body the same way as a firefighter. Firefighters wait in the office preparing materials or doing paperwork, more easygoing work, until suddenly they get a call about a fire, and they immediately leave the office and rush to the scene of the fire and put out the fire as quickly as possible. In this sense, factor 8 is the firefighter, and another biological molecule called factor von Willebrand acts as a fire station. When these 2 biological molecules are bound together, factor 8 remains inactive, but when it detaches from factor von Willebrand when it hears news of bleeding, factor 8 becomes activated and it rushes down the fire pole, or in this case the bloodstream, and goes to the site of the blood and tries to clot the blood as quickly as possible to prevent any damage. In hemophilia A patients, imagine a bunch of firefighters using water bottles to put out a wildfire. There just isn't enough factor 8 secreted to stop the bleeding. This seems really terrible, and it is, but one thing that's helpful about this disease is the fact that it's a single gene mutation, meaning that in theory, if we use new and improved gene editing techniques to remodify the gene to its normal form, the symptoms of hemophilia A should be corrected, as the factor 8 protein should be produced by the liver in sufficient quantities, greatly improving their quality of life. There are many ways that treatments are being approached by biotechnology, but I focused on using TALENs and induced pluripotent stem cells. CRISPR is the most well-known gene modification technique right now, and for good reasons. It's simple, affordable, and easy to use. But CRISPR techniques for hemophilia A have only been tested in mice so far, and there are a ton of risks and complications that occur with using CRISPR in the moment. And eventually it could be a feasible technique, but for right now, Dr. Wu outlined the use of iPSCs and urine cells using TALENs, and this was a successful treatment. Induced pluripotent stem cells are super cool. For any Harry Potter fans, it's like cells taking polyjuice potion. When engineered properly and carefully, they can shift any body cell, usually blood or skin cells, but anything works, into embryonic cells that can be used for any purpose. Since these unspecialized cells can essentially become any other cell, they're super useful for drug development, gene therapy, regenerative medicine, and more. Going back to TALENs, which stands for Transcription Activator-like Effector Nuclease, this technology uses engineered enzymes to cut and target specific sections of DNA. There are two parts, a DNA binding domain and a DNA cleaving domain. The DNA binding domain acts like a finding tool in Google Docs. It matches a sequence of base pairs to a certain section of DNA and binds at that location. The DNA cleaving domain acts as a delete button, cutting the DNA at the location that the DNA binding tool identified. TALENs have a better range and are more effective than the first type of gene editing called zinc finger nucleases, but are still more expensive and difficult to use compared to CRISPR. For the process, urine would be collected from a hemophilia patient, not blood because that could be dangerous, and the F8 gene mutation in the DNA extracted would be found using inverse shifting polymerase chain reaction. The urine cells would then go through a lab process to become induced pluripotent stem cells. Keep in mind that eventually these processes would be happening to actual body cells or even embryonic cells when the safety standards and ethical standards are met. Two pairs of TALENs would then be used to target the intersection of exon 22 and intron 22. In this study, they used L1R1 and L2R2 as the TALENs. Then the target section of the gene would be amplified using PCR and gel electrophoresis. Since the inversion is the majority of the gene, it's easier to shift exons 23 to 26 to the correct location than to move exons 1 to 22. Therefore, exons 23 to 26, along with an SV40 poly A signal and a PGK neocassette, are put together to create a donor vector. Finally, the DNA is cut and rejoined using homologous repair, and the process is complete. In this experiment, the results were incredible. It showed that the transcription of inversion 22 was corrected and that the protein was now being secreted in proper amounts. The scientists also checked for off-target effects, which is when the DNA targeted section can be present in another section of the DNA, which is a main concern for gene editing right now. Along with off-target effects, scientists also have to be careful of the iPSCs undergoing rapid mitosis, which could result in a tumor. And along with that, the DNA can also be mutated during homology-directed repair. So there are a ton of safety standards that have to be met before this treatment can be sold in your local pharmacy. We may not think about some of these genetic disorders all the time, but millions of people around the world are impacted and suffering from these diseases every single day. It's crazy to think that an entire human body can be disrupted for a lifetime because of a mistake made by your DNA while it was copying. But everyone, including your DNA, makes mistakes. And if we have tools to combat these errors and try and fix these big problems, it's our job to work hard and try and reach a future where all genetic disorders, once thought to be untreatable, are all treated and no longer exist. Thank you.